to introduce Craig Forrest. I met him, I think, for the first time about an hour ago, but I may have met him many years ago at MIT. He did his PhD at MIT and overlapped actually with both Andrew and Brian at MIT. But he's currently from Georgia Tech and has become very involved in human brain recording. He's going to tell us about automated patch clamping. He's also spent time at the Allen Brain Institute, and one of his other strings to his bow is being involved in developing makerspaces. I think that's the primary reason you came down here, wasn't it? Just to help with the design of the makerspace that's been opened in engineering that was launched last week. So welcome, Craig, and we look forward to your seminar. And I think there's quite a few people here that are involved in electrophysiology and interest in patch clamping, so I'll be very interested to see you.
Okay, terrific. Thank you for the kind introduction, Peter. It's really great to meet you. For those who don't know, I did my PhD with Peter's brother, Ian, and so it's great to sort of come full circle and meet his brother and see my old colleagues here. So it's a pleasure to share with you some of the work we've been doing in my lab at Georgia Tech, in particular trying to measure the electrical activity of single cells in the living brain, but also measuring other characteristics of those cells, such as their shape, how they're connected to their neighbors, and even their genetic information. There's a gold rush on right now in neuroscience to try to develop new tools that can do these things in the living brain, to try to understand how the brain works. So it's a pleasure to tell you about that and show you some of the things we're up to. So, you know, this search for how the brain works has been going on for a long time, even back in the 1800s. Probably one of the most famous neuroscientists, Santiago Ramon y Cajal, looked into a microscope and for the first time saw the shape of neurons in the brain and drew these pictures, fantastic pictures showing the axons and dendrites, the pyramidal shape of these neurons, the layers that they were in, and somehow that the cortex, the outermost layers of the brain, had this sort of regular pattern of circuits that provided some computational ability that we still don't understand, but that's really what makes humans unique, is this massive surface area of these cortex with these columns, these columnar circuits across the cortex. So it's of great interest in neuroscience to understand how neurons communicate within the cortical layers of the brain. So a couple years ago, President Obama personally championed a big new initiative for neuroscience, and that was called a BRAIN initiative. BRAIN is an acronym. And lots of engineers like myself thought this is a gold rush. We've got to develop new tools to understand how the brain works. So no longer do we only get funded for hypothesis-driven research, but now just developing tools is worthwhile for its own sake. So it's a great time to be in the field of neuroscience and neuroengineering. And our lab was involved with this from the beginning through this technology I'm going to share with you shortly. One of the key papers for the BRAIN initiative is this one, if you're curious. This is really a summary of the entire multi-year effort. It's currently around a $500 million effort in the US, and this paper at the time was called the Brain Activity Map, but subsequently just called BRAIN. And there's a couple key passages that I pulled out from this paper here to summarize it. Okay, so this is really the motto for our lab. You know, new directions in science are launched by new tools much more often than by new concepts. Okay, the effect of a concept-driven revolution is to explain old things in new ways, while the effect of a tool-driven revolution is to discover new things that have to be explained. I mean, imagine the first time you turn your telescope to Saturn, you see all these moons. Now you got to figure out how the moons work. How did they get there? So it's the telescope that enables that new science. But the state of neuroscience today is pretty primitive, and I pulled a quote also from this paper on the right side here that explains the state of neuroscience today. Okay, to explore these jungles, meaning these tangled networks of neurons in the cortex, Scientists have traditionally relied on electrodes that sample brain activity only very sparsely. Okay, it's like, and what it says here is it's like watching TV and you can only see one pixel. I mean, imagine that show is not very interesting. You know, what's the show all about? What are these characters talking about? And you can only see one pixel flashing. So we really need the ability to record from many, many neurons simultaneously and to see how they're connected and to understand how these neural circuits work. So this is like the holy grail of neuroscience right now. So I've got a little video of what we've been up to, and hopefully you'll see how it relates, and then I'd be happy to take some questions. Oops, pardon me.
The brain is a complex circuit made out of hundreds of billions of cells called neurons, each of which communicates with thousands of others. Each neuron receives information from upstream neurons, computes an electrical signal within it in milliseconds, then triggers the transmission of information to downstream neurons. These neurons are heterogeneous. They express many different sets of genes and have many different shapes, and they change in different ways in different brain disorders. In order to understand how the neurons compute, ideally one would be able to observe individual neurons processing electrical signals in the living mammalian brain. And ideally, one could derive a complete list of the different types of neurons that make up the brain. By seeing how each type of neuron changes in a given brain disorder, one could identify molecules that distinguish unhealthy cells from healthy ones, which could then serve as better targets for novel drugs that have fewer side effects. A team of mechanical engineers, neuroengineers, and neuroscientists has now developed a robot that can analyze the electrical and molecular properties of single cells in the living mammalian brain. The robot consists of a glass microneedle known as a pipette, with a tip just a micron wide and filled with electrically conductive fluid. The pipette tip can be moved around by a precision motor. The robot docks the pipette to a single cell in the living brain so that the pipette can capture electrical signals and molecules from the cell. The electrical signals are transmitted up the pipette, amplified, and sent to a computer. The molecules can be harvested by extracting the contents of the pipette. The procedure of using pipettes to record neural activity, known as patch clamping, has been performed by humans for decades. But in the living brain, it is difficult to do, something of an art form that only a small number of people have mastered. This robot is controlled, in contrast, by an algorithm, a piece of software that enables neurons to be detected, electrically recorded, and molecularly harvested in a high-throughput fashion without requiring human intervention. The robot lowers the pipette into the brain, looking for a cell to record from. Pressure is applied to the inside of the pipette so that it constantly ejects a small amount of liquid to keep the tip clean. It detects cells by delivering small pulses of electricity while the pipette is lowered in small steps, each about 1,000th of a millimeter long. When no neuron is present, the pulses of electricity diffuse away into the brain. When the pipette is close to a neuron, however, the neuron blockades the flow of electricity. The software running the robot monitors these electrical signals over time. This software, in essence, equips the robot with a bit of intelligence, enabling it effectively to see a cell when it is nearby. When it detects the blockade of the flow of electricity, indicating a neuron to be present, the robot halts the motion of the pipette. Then the robot applies a bit of suction to the cell, bringing the cell's membrane or boundary in close contact with the pipette tip. This is crucial because it is important to ensure that the pipette and the cell are touching closely. Finally, a brief pulse of suction results in the rupturing of the cell membrane, enabling the inside of the cell to be connected to the inside of the pipette. Now, electrical activity within the cell can be recorded with extraordinary precision. Also, if a dye is contained in the pipette, it can diffuse into the cell, enabling the shape of the cell, an important determinant of how a cell computes, to be seen. And because the pipette has access to the inside of the cell, it can harvest the contents of the cell body, which can be pulled into the pipette with a bit of suction. In this way, the robot can perform many kinds of observation, electrical, molecular,
All right, cool. I brought along some pipettes, in case you haven't ever seen one of these. And so the 1 micron tip pipettes, there's some borosilicate and also fused silica ones. What questions do you have from that video?
Can you do multiple plunging?
Oh, good question. Yes, we sure can. I've got a couple pictures of that later. So if I can come back to that, I'd be happy to show you. Yes, sir.
The video tells us that a suction was applied to get to the cell.
That's
In
right.
your original paper, I think you said that you were using ZAP. I wonder why you've changed from ZAP now to. You find it more effective, or is it easier to perform, or?
Right. So in the original paper, which this video is a summary of the original paper, I believe we used a suction. So the pipette stops abruptly when a cell is detected and then applies a bit of suction. For those who've done patch clamping, zapping can be helpful in terms of temporarily opening pores in the cell to gain access. But we actually don't do that. We do not do zapping.
Zapping is just high voltage.
Right, exactly. Yes, sorry. It's like electroporation.
Yeah, and the video shows neurons, but there's also glia cells. How often do you?
Oh, good question. Good
do you
question.
touch on?
Yes, so people are not really interested in the electrophysiology of glial cells. They're not electrically active. So of course we do find some of those. We get them about 10% of the time, 10%, and that's less than you would expect by chance. And we think the reason, of course it's hard to tell why, but we think the reason is that the positive pressure we apply pushes them out of the way. So they're not as large and they're not as well connected to the surrounding tissue, so they move out of the way under the positive pressure is what we think is happening. So we get them about 10% of the time. Yes?
You mentioned injecting a fluorescent tracer. How do you actually read that out?
Oh, great. Yeah, that's a great question. So people are very, very interested in how the electrophysiology of cells corresponds with the shape of the cells. And so ideally, you'd be able to do both of those at once. So there's a couple techniques that are very popular. One is Bioengineering staining. So in that case, you have a protein that embeds itself in the membrane. And then you add, after post-recording, you fix the tissue, and then you stain it with a labeled streptavidin dye. Okay? And then you can see in a simple fluorescence microscope in a slice of tissue, the cell. The second and a much better way is to diffuse a DNA plasmid across the membrane. And in this case, if you retract the pipette very carefully, you can get the cell to heal up and survive. And then about 3 days to a week later, you can sack the animal, and that cell will be expressing its own dye, GFP typically. And that's actually far superior because the labeling is not based on diffusion, but since it's expressing its own fluorescence, you can actually get expression all the way at the very most distal ends of the cell, which can be millimeters or more away.
But it's not exactly high throughput then to find the same cell.
To find the same cell?
Yeah.
You mean after post-recording? Right, that's not high throughput, right. So you complete your experiment. So let's say you record from 10 cells in the animal. You stop, you sack the animal, and then you slice the brain very finely, and you take all those pictures. Right, so that does, of course, take some time. Absolutely. Yes? Sorry, in back. And then we'll come to you next.
How deep have you gone down? What part of
Yeah.
brain have you studied so far?
Okay, great question. So we have advanced this technology over the past few years to the point where we can now do anywhere in the mouse brain. The mouse brain is about the size of a peanut, and the center of it is about 3 millimeters deep. So we can get there. And the key to that, which I'll talk about a little bit later, but I'll give you the punchline now, is avoiding the blood vessels on the way down. If you impale a blood vessel with your pipette, you ruin the tip. So we discovered that we could detect the presence of a blood vessel based, again, on the resistance, stop, and go around that blood vessel. And I'll show you some data. So historically speaking, thalamic recording is almost impossible with patch clamping in vivo, and we can now do it with a pretty good yield, about 20% yield, okay, which is pretty good. Yeah, okay, and your question, sir?
This one related about navigation system. How do you guide the tip of micropipette?
Yeah.
In the focus location?
Okay, good question. So guidance. So what I've just shown you here is completely blind. completely blind. So what you're doing is you're saying, I'm interested in the hippocampus today. The hippocampus starts about 1 millimeter down in the mouse brain. So what I'm going to do is I'm going to rapidly traverse through the first millimeter because I'm not interested in that. And then I'm going to start hunting in that region. And I'm just going to take the first neuron I find. So that's a lot different than saying, I want this particular neuron here. Maybe, you know, a layer 5 pyramidal cell. That's exactly, I only want that, right? So there are some solutions to that. The most popular one is to use a transgenic mouse, which has fluorescent neurons of the type you want, let's say layer 5 pyramidal neurons. And then what you do is you use a 2-photon microscope to visualize those cells below the surface. And then you can steer the pipette to those cells. Now, so far that has only been possible manually, but we just had a paper accepted in Neuron showing closed-loop control of that process. So you can imagine it's like a heat-seeking missile, right? You want to click on the cell, and then it just does its magic, and the pipette lands on that cell. And we just recently got that to work. And this is in collaboration with Ed Boyden at MIT, and we just published a paper in Neuron where his grad student Ho-Jun Suk is the lead author. That's a fantastic piece of work, yeah. Yes?
Maybe one more. Your video shows an ideal situation that your target is static, but as we know, particularly if you go at the surface regions of the cortex, your target actually is moving
yes.
up and down due to pulsations of the brain related to heart rate and respiration.
Right.
I wonder how this signal, this movement, interferes with detection of the cell and decision where to suck
yes.
or not to suck.
Were you one of the reviewers on this paper? Because that's exactly what they asked. It's a big problem. It's a big problem. The reason why people prefer to patch neurons in culture or in slices ex vivo is because of micromotion. So in vivo patching is notoriously difficult. Most people who've done this for years, if they're very good, they can patch 1 or 2 cells a day. That's how painstaking it is, how low the yield is, and in large part because of micromotion. So we have looked at this problem very carefully, and we can see the heart rate as sort of a carrier frequency on the electrophysiology signal, and the respiration. The bottom line is our yield is around 30%, which is about 50% better than the best humans. So we haven't completely overcome that problem, but the robot can do it better than a human. You could imagine in maybe a future generation of this technology if you can somehow move the actuator in time with the heart rate or the breathing, and then sort of cancel out those motions, but we're not there yet.
It's 400 beats per minute, the heart.
Yeah, pretty fast. Mice have pretty fast heart rate.
It's so easy to follow this actually.
Yeah. Let me go on to a few other slides, and I'd love to take a few more questions a little bit later. This is what the data looks like. So any sort of hardcore neuroscientist, they look at this, they say, oh, that's a beautiful recording, very low noise. And then when you inject current, in this case we're injecting 80 picoamps of current into the cell, you evoke action potentials. That's what neurons do. That's how they talk to each other. Out here in this kind of noise floor here, what you see is this is actually physiological signals here. These are called subthreshold up-down states, meaning the neuron's not spiking, but it's got this sort of jitter going on, and that's characteristic of in vivo recording. So of course our neurons are all connected to each other, and when the neurons around you are firing, this neuron is going to get some inputs, and this is the sort of summation of inputs to that neuron, and the up-down states are much higher amplitude than in the hippocampus. This is a characteristic of pyramidal neurons. So we started a company around this technology a couple years ago called Neuromatic Devices. They produce the pressure control box and the associated data acquisition equipment that allows you to convert a manual patch clamping rig into an automated patch clamping rig. And they've sold those all around the world now. This is what kind of the full-up machine looks like. So there's kind of this server rack with the amplifiers and digitizer, motor controller, and this special box by Neuromatic Devices. And then on the business end, you've got what's called a head stage. That's a little preamplifier attached to some actuators and a vibration platform. We've distributed the software freely on autopatcher.org. Hundreds of labs have downloaded the software and built their own machines. So we provide all the instructions if you're more of a DIY type, or you can, of course, buy the hardware from the commercial outfit. And just full disclosure, I don't have any equity in that company or any compensation from that company. So our lab produces the software. These are just different versions of the software for different hardware configurations that people have produced. So we produce the software in my lab, and the company produces the hardware. So you asked about multiple, so I'd love to show you more about that. So this is one with 4, and then we built one with 6, and this is all in vivo, and we performed the first ever quadruple simultaneous in vivo patch clamp recording. It's extremely difficult to get one manually. You can imagine getting 2 is pretty much impossible. I think there are no published reports of manual dual patch clamping. There is a couple where you have one patch clamp and one what's called a cell attached recording, which is like a loose patch clamp recording. But here we have 4 high quality recordings. What this does is allow you to measure the connections between neurons, which is very important. And we're working with Emory Brown at Harvard to study the effects of anesthesia on different cell types. So imagine you can patch clamp onto different cell types simultaneously. So one excitatory neuron, one inhibitory neuron. Then you deliver anesthesia to the mouse and you see how they change differentially. No one really understands how drugs affect different types of neurons and different types of ion channels. Even large drug companies like Pfizer in Cambridge, who we've been working with, are doing their ion channel measurements in slices of brain tissue. They do not do them in vivo, and things are much different in vivo. When they do in vivo work at Pfizer, they only use extracellular recording. They do not use the high-resolution intracellular recording because it's so difficult to do. So other things we've done with this technology over the past few years since we invented the auto patcher is to miniaturize it. That allows us to pack in actuators more densely and reduce the cost. And we've also miniaturized the electronics. So we've worked closely with a company called Intan Technologies through a small business grant with the NIH to miniaturize this. And just to put this in perspective, on that server rack I showed you before, there was about $25,000 of amplification and digitization electronics. This is a Symonds fabricated board here with which retails for $5,000. So it's currently for sale by Intan Technologies for $5,000 instead of $25,000. So that's where that is at. And the signals are quite good. Of course, there's a typical challenge with reducing electronics down to CMOS scale is noise platforms. Of course, we thoroughly characterized the noise, and it's quite good. It's certainly good enough for whole cell recording. But to be frank, if you're interested in doing this, it's not quite good enough for single ion channel recording, which is something else people typically use this technology for. Let me tell you just a little bit more about the multi-patching. So we recently received a grant to study long-range connections in the mouse brain. And one of the ones that's most interesting to neuroscientists is this pathway called the Thalamocortical circuit. So there's a highway in our brains between the thalamus, which is like the primitive part in the center, to the cortex. And there's different highways for different functions, but this one is for sense of touch, okay? So what we can do is we take a mouse and we can flick the whisker with a piezoelectric actuator, and we aim to measure the connections between the thalamus and the topographically aligned neurons in the cortex. So it's kind of like, I don't know if you ever heard of the homunculus. It's a mapping of our sensory system in the brain. So it's beautiful in the mouse. The whisker pad is exactly represented on the surface of the mouse brain. So it's topographically aligned. So we've integrated these multiple pipettes with a camera system to detect the right whisker on the surface of the brain using a technology called intrinsic optical imaging. And this is what led us to discover that we could dodge around blood vessels, as I mentioned earlier. So when the pipette goes into the brain, if it hits a blood vessel, the blood vessel deflects until the pipette punctures straight through it. And when it does that, the resistance initially increases and then returns, but it doesn't quite return to the baseline. There's a bias. That's because of the fouling of the tip. Those pipettes that are being passed around, they have a 1-micron tip, and any junk that gets into that tip will prevent sealing onto a cell later. So we discovered that we could we can dodge these blood vessels. So you see, when you get the robot controlling everything, you can start to make it smarter and smarter, and now it's flying through the brain. Okay, when it bumps up against a blood vessel, it can back up and go around. That's what is depicted here in this cartoon. And it does a sort of spiral search pattern, which is the most efficient way mathematically to find the way out around the blood vessel. So like if you're in a sailboat, if you're in a rowboat and it's foggy and you can't find the shore, your best bet is to do a logarithmic spiral pattern. Make a note in case. So using this robotic navigation to steer around blood vessels, we've been able to record, these are the highest quality thalamic recordings ever produced, and they look just as good as a traditional in vitro thalamic recording. In vitro is when you cut the thalamus out of the brain and put it on a dish. This is in vivo, okay? So the way that you can tell these are good is the membrane potential is very steady at about minus 55 millivolts. The overshoot on the action potentials is above 0 millivolts. And this has characteristic, for the electrophysiology gurus, it's got the characteristic features of the thalamus like this sag current and this rebound spiking and so forth. So we don't do the neuroscience in my lab, but this is the kind of data that gets neuroscientists excited about the kind of things they can study now with this. We can also do very long recordings. It's almost impossible to record from many, many minutes in the thalamus in vivo, but we've been able to do it with this dodging. And so what this means is there's no leakage current. The seal is very stable because the pipette was clean when it hit the neuron. So we can perform very long, when I say long, about an hour, okay? And you may wonder, why is it only an hour? Well, remember, the pipette is connected intracellularly, so the contents of the cell are kind of diffusing out into the pipette, and that the cell's not happy about that, so will eventually die if you record too long. It's not like extracellular recording where you can record from a cell for even days. So putting this all together, recording from the cortex and the thalamus simultaneously, while flicking the whisker, we can measure the delay in the action potentials as that signal propagates through the circuit in the brain. And this is the kind of stuff the neuroscientists are drooling over this, because it, for the first time, allows us to measure the strength of these connections, the speed of these connections, the probability of these connections. This is not known at all, okay, this kind of data. Let's talk about filling a little bit. You asked earlier about what are the different ways to record the morphology of the neurons. So we've recently added a new feature to the autopatcher that allows it to automatically do the morphology of the cell. And let me explain how that works. So the autopatcher is doing its business. We locate it wherever we want in the brain. Let's say we want to record from the thalamus today. We hunt for a neuron there, again using that stepping you saw in the video, applying suction to seal onto the neuron. We record from the neuron to make sure it is in fact a neuron and not a glial cell. And if we're in business, so we see action potentials, now we can start to do the functional cell type characterization. So in this case, what we're doing is the mouse is looking at a computer monitor, and we show drifting gratings. This is a classical drifting grating test to characterize the cell type functionally. And then what we do is we can electroporate that cell and deliver a plasmid and then retract the pipette. We've now got this fully automated. Of course, animal recovers, and then we can slice the tissue and image the cells. That's what I was saying earlier. Let me show you some of the pretty pictures that result from this.
So you didn't go for cell in this case?
That's correct.
Cell attached.
That is correct. That is absolutely correct. This is a cell-attached recording. It's not as high quality. That's correct. Very important qualification. So in this case, we've reconstructed the various processes of the cell, and here I'm showing it alongside the visual stimulation and the evoked potentials from that. Okay, this paper was recently published as well, and it allows us to catalog neurons along multiple axes. Okay, so you can say cells that fire like this look like this. No one's been able to do that before in vivo. It's incredibly difficult to do this. See, we don't even know what types of cells are in the brain. This is just, if you want to know how a car works, you start by, looking, okay, it's got 4 wheels, it's got these lug nuts, it's got this engine. We're basically just cataloging the types of cells. So groups like the Allen Institute for Brain Science in Seattle have teams of technicians that are just cataloging things. They have recently unveiled a mouse atlas, they call it, which has thousands of neurons they patched in the visual cortex. So you can see some more of our electrophysiology results here. The contrast is a little poor, but you basically can see how beautiful the reconstructions are using this GFP. Now, the next thing that makes patch clamping really, really hard is after every single recording, you have to throw away that glass pipette. So this technology was invented about 40 years ago. They won the Nobel Prize, patch clamping. And every single recording that's ever been done is a single-use disposable pipette. It's a big pain. And it's especially a pain if you're trying to automate it, because although the robot does everything, you still have to sit there and feed it pipettes. Okay, so we started wondering, is there any way we can clean the pipette and reuse it? Okay, so everybody says this is not possible. Hamill, in his Nobel Prize address, Edwin Neher, this is his Nobel Prize address in 1992. Make sure you use a fresh pipette for each approach, okay, and so on with the other founders of this technique, okay? So we were recently surprised to learn that we could in fact reuse the pipettes, okay? So what we figured out how to do here is after the recording, we move the pipette into a cleaning basin, and then we suck up and squirt out some, it's an alkanox, basically a mixture of surfactant and water softener that allows us to clean the pipette, and then we rinse the tip in artificial cerebral spinal fluid, and we can go back to the cells. So this process takes us about 76 seconds to do, and let me show you a video of it in action, okay? So this is the first ever serial, fully automated patch clamping. So every time this thing lights up green on the lower left, the LED, there you see that green LED? It's got a cell. Isn't that great? You don't even have to be there anymore. You see, the engineers are done when you can just turn off the lights and go home and it just gets all the data for you. So you can imagine when the Allen Institute has like a room full of technicians doing this all day, like this is incredibly valuable, right? Just load one pipette, click on all the cells you want, and then leave. Now, these cells are in culture. They're adherent, so it's a bit easier. I've cheated a little bit, okay? But let's see what we can do here. So going on, okay, this paper was recently published in Scientific Reports. Here I'm showing you some different cell types. This is hex cells. This is all done with one pipette, these hex cells, and then a second pipette was used to do these neurons in culture. A third pipette was used to do neurons in brain slices, and here a fourth pipette was done doing neurons in vivo. Okay? So being able to reuse pipettes is incredibly useful for automating patch clamping. So what about when you put this all together in brain slices and in vivo? Now you've got a new problem to deal with. Let's say you select your 10 cells and you release the machine, like go get those 10. The problem is as the pipette goes in, it physically displaces the tissue. So the cells move out of the way. So now you really wish you could track those cells as they move around. So you see when the pipette ends up next to the cell's not there anymore. So you obviously can't patch it there. So we've been working with a collaborator at Georgia Tech named Chris Roselle, who's in electrical engineering, to track the cell position using microscopy and then to correct the pipette position so that it gets that cell. And I'm pleased to show you a very cool video. Here's 10 neurons in a slice. This is not in vivo, but in a slice, which were all patched with a single pipette without being in the room. Okay, and yeah, so the video is not much to see because it's just a pipette coming in over and over again and hitting that tissue. But this is spatially what happened. Okay, so I think it missed 2 of them, maybe the red ones. So you got 8 out of 10, not bad. And you can see the electrophysiology from all those. So again, this is the first time anybody's ever patched multiple cells in a brain slice without being there, and it really allows us to do dramatic increase in throughput. Okay, so now you might wonder back about multiple pipettes. So I've got a video about this. So now we've got 2 pipettes that are both working in coordination with each other, cleaning and hitting the cells over and over again. So now we've got on the right, you've got your rig with the 2 pipettes. Of course, it lights up green when it's got a cell, and they're cleaning themselves and going back each time. And in this case, you can see the counter on the right. So whole cells, we get 4 out of 4 in 11 minutes, and let's keep continuing here. We always have the empty chair in the picture. That's very important. So just to calibrate your expectations here, for a really good in vitro patch clamping person, they might get 5 or 10 cells for the whole day for themselves. This robot here got this in about 27 minutes, no unattended. So we go through a lot of pipettes in the lab, so we made a machine to make pipettes. And I've got a video of that. This is kind of fun. So I think for my colleagues from the bioinstrumentation lab, this really is our bread and butter here. Of course, we got your Arduino with the touch screen. We've got your extruded aluminum, your Festo actuators. So this is using fused silica, which is on a spool. It's coated with polyimide, so it's flexible. And the challenge of using fused silica is the melting temperature is extremely high. So we actually use a gas flame here, but commercial pipette pullers for fused silica use a CO2 laser. So this is all running autonomously here. You'll see in a second what happens. So the pipette is fed in with these rollers, left to right. It's clamped. It's under tension here while it's being heated. And then it necks and pulls and breaks the pipette.
Is it what you had in your box next to
Yes, that's right, exactly. And then those drop into a little gel pack here on the lower right. And so, yes?
How does the repeatability of the arc?
With the flame, it's not great, to be honest with you. We actually got contacted by a company that makes pipettes for in vitro fertilization. Those are a lot easier to make. They're huge, relatively speaking. Actually, did you know the world market for in vitro fertilization pipettes is about 10 million pipettes a year, and they're all made manually. There are companies in like Eastern Europe that do this, and they're manually pulled and polished, hand polished at a bevel. The bevel is for puncturing the cell, and then the other one's a holding pipette. Anyway, so we're this could be good enough for in vitro fertilization, but frankly, it's not good enough for patch clamping. The patch clamping.
Why not use electrical heating?
Yeah, so you can do that with the borosilicate. Okay, the borosilicate pipette pullers use a nichrome wire, electrical heating, but the fused silica requires over 1,000 C, which is not possible with nichrome.
So everybody thought until recently, obviously, that you need a glass. Glass has got this special properties which seals to the bilipid membrane.
Right,
forms
right.
this famous gigaseal.
That's right.
Do you get gigaseals also with silica?
Fused silica, yeah, also glass.
Fused
It's
silica.
quartz, the same as quartz, yeah.
So it is glass,
Yes,
not the glass. Okay.
yes. It's coated with polyimide, that's why it's orange.
I was wondering why it is.
Yeah. If the glass is not coated with that polyimide, it's incredibly fragile. The polyimide keeps it from breaking when you bend it. But of course, the flame burns that off. And then we made a machine to inspect the pipettes. Of course, you want to make sure the pipettes have the right size. So people train to be able to look at a pipette and know it's the right shape, but that seems kind of painful. So we made a machine that does that for us. You load the pipette into it. It automatically focuses it. As you'll see here, the pipette is snapped in to a little kinematic fixture there, and then the operator just clicks start, and it finds the tip using an efficient search algorithm, and then it focuses on it, and then it processes the image to find the, critically, what people really want to know is the diameter and the cone angle. That's what the electrophysiologists really care about. So that concludes my talk. I'd love to take some more questions from you and acknowledge my students and collaborators, predominantly the Allen Institute for Brain Science in Seattle, Washington, and Ed Boyden at MIT, longtime collaborator. I'd be happy to take some more questions that you might have about this. Thank you very much.
So what's next?
What's next? Yeah. No, we're done. No. Yeah, the next thing.
Viral injection?
The next thing is we're working with, I mentioned Ho-Jun Suk from MIT. So he's got this closed loop 2-photon guidance. So there are certain neurons that people think, they're called interneurons. People think they are the modulators for the signal in the electrical circuit in the brain. So the interneurons are small and they're rare. So the odds you're going to hit one by chance is pretty low, especially when you only have a 30% yield to begin with. So you have to patch over and over and over again with a 30% yield to get one of these interneurons. So what we've been able to do with Ho-Jun is, using transgenic animals, target, using 2-photon microscopy, target fluorescently labeled interneurons in vivo. And then what we want to do is we want to couple that with the multi-patching and cleaning technology to patch neurons that neuron is connected to study the synaptic properties of neurons. There are some hypotheses out there that a neuron's identity is conferred by its neighbors, who it's connected to. It's like how if you meet all of someone's friends, you know what the person is like. So we've got to meet all the neurons' connections. So that's a kind of synaptic patching bot that we're working on. And the other thing we're using this technology for is actually applying it to something called brain organoids. So stem cell researchers have recently been able to grow miniature human brains from patients, human patients. And the way you do it is you take the blood out, take some blood from the patient, you separate out the white blood cells, you revert those white blood cells to stem cells, induced pluripotent stem cells. Then you can derive them into neurons. And you can actually grow a miniature human brain about the size of a pencil eraser. It's fantastic. Okay? So people want to verify that these brain organoids, they're called, organoids, have the same properties as somatic tissue, adult tissue. Okay, so what we want to do is be able to take the multi-patcher and put it inside of these brain organoids and measure the connections. So that's a sort of more scientific endeavor that we're collaborating with some folks at Emory University on that. Yes?
Have you used this technology for sharp microelectrode recording?
Sharps recordings, right. So we have not done that in our lab. A lot of people do those types of recordings. There's a couple challenges with it. There's also a couple of advantages, but there are a couple challenges. One is you can't record very long. So the way Sharps recording works, for those who don't know, is you basically make a pipette very sharp, actually about 1/10 of the pipettes I passed around, only 100 nanometers in diameter, and you just stab it into the cell. So there's no gigasealing. Problem is the cell is sort of leaking its contents, so you cannot get a stable recording over the long term, especially in vivo. It's very difficult to do. But we are doing, I'll tell you one project we're doing on that front. So some of our collaborators at the NIH in Bethesda, Maryland, they are trying to grow retinal pigment epithelium from induced pluripotent stem cells as a therapy, a cell therapy for macular degeneration. Right now, if you have macular degeneration is the leading cause of blindness in old people, there's no cure. These guys think they have a cure. What they do is they take stem cells, they derive them into retinal pigment epithelium, and then they grow them on a biodegradable disc of PLGA, and then they insert it underneath the retina. They've done it in pigs and restored vision. It's super cool. This year they're going to do 12 human patients in a clinical trial. And we are involved with that by testing out the electrophysiology of these cells to make sure they have the same properties as normal RPE cells, retinal pigment epithelium. So when you're trying to get FDA approval on a drug, you have to test its efficacy within 1 week of implantation for a cell therapy. So that's why we're going to sort of certify that these cells are electrically active. And that's a Sharps recording. So in that case, we are just impaling the cell.
Why do you need to use Sharps for that?
Why do we need to use Sharps for that? That's a great question. The RPE cells are different from neurons. They're tight, they have tight junctions, and they're in a monolayer. So you can't seal on them very well, basically. They also have these distal processes. They have like this carpet over the top of them, this hair. So you can't really seal onto them. They're not like a smooth basketball. So traditionally, you just stab them. Yeah. Yes?
So the pipettes were advancing very fast, which is really impressive. But have you thought about the mechanical changes that could be induced? So can you change the rate of advance when the pipettes are coming down into the cell? And if you do change it, if it's slower
Yeah.
or if it's faster, are you actually seeing different properties, passive and force-fighting properties of those cells?
Right, right. No, that's a good question. People ask us all the time, can you optimize the robot for my favorite cells? And I think there's a big opportunity there, but we haven't. It's very difficult to do these experiments, and so it's hard to sort of systematically scan the parameter space. Like you can imagine all kinds of knobs, right? What's the best pressure to use? What's the best speed, as you say? What's the best voltage? And when you're trying to detect the resistance. And it's hard to optimize, but it's probably true that there is a different optimal speed and pressure and so on for different types of cells. We just haven't, we're just using one now because it seems to be working pretty well. You know, biology is kind of fuzzy like this. It's real, I mean, you guys are, for mechanical engineers like me, it's very frustrating because there's such a wide variation in types of cells. It's difficult to be precise about it.
Yeah, just wanted to, it seems to me to be really lovely with the whole brain work to have this a little bit more directly linked with imaging. And
Yes.
the photon is both restrictive and expensive
Yes.
as an option. Is there any work going on in using optical fibers to actually look more closely at what's going on at depth?
I'd love to do something like that. It's a really cool idea. One person actually suggested once to use the pipette itself, which is a cool idea. I mean, the pipettes are actually made out of the same glass as optical fibers. So you can transmit light down the annulus.
Well, I guess the optics wouldn't necessarily be great.
Right, right.
But I mean,
Here's the best we've done in this front. We have taped an optical fiber to the side of the pipette
yeah.
and gone in, and after we patch clamped, we can turn on the light and verify that the cell we're recording from is one of the cells that was genetically engineered to light up, to turn on with light.
Yeah.
So in that way it gives a little cell type specificity to the patch clamping.
Yeah.
But it's still blind targeting. It's just, this is a real, optical access to the brain is a huge problem.
Yeah.
Okay.
Sure.
Even for the lowly mouse, you can't see the entire cortex with 2-photon microscopy. There's some promise about 3-photon microscopy getting down the full cortex, 1 millimeter, but it's still an active research area. It sounds like a great idea. Why can't I just read out all the electrical signals with light? But in practice, getting that light out is hard. Getting it in and out is hard.
I mean, the other thing is also that the light's only going to give you a certain amount of information in terms of functional imaging.
Right.
Beyond that, so you're still, in terms of biophysics, you've got to have access to the intracellular.
Yes.
domain.
Yes. So this is one of the dirty secrets of optogenetics as well. You cannot record subthreshold activity with anywhere near the resolution I'm showing here. So both in terms of the time, the frequency of the signal, we're doing 10 kilohertz here, and the subthreshold resolution. Patch clamping is still unparalleled. So while it's a old technology, it still is the best technology for recording from single cells deep in the brain. I mean, I hope optogenetics will replace it. I really do. It's a pain.
Well, you don't, I mean, it seems to me that those two things might be integrated.
Maybe, yeah.
You know, that functional optical imaging
Right.
will give you some information which might add to what you can also get with single or dual patch clamps.
Right. For example, there have been some experiments where people have patched onto a cell and then turned the neighbors on with light.
And then see what.
Exactly, see what's connected to.
That's right.
Yeah, so there's an integration. And the project I mentioned again with the 2-photon patching interneurons, that's another example of integration. So yeah, if you look at the Brain Initiative projects at the NIH today, there's sort of 3 categories of things. There's probes, like what I showed you, and also other kind of probes like carbon fibers and silicon probes. Then you have your molecules, new kinds of voltage sensors, new kinds of neurotransmitter sensors. And then you have your microscopes. Probes, molecules, and microscopes. And the microscopes are things like in vivo microscopes, little tiny microscopes you can put in the brain, or 3-photon microscopes, or wide area microscopes, high resolution, large area. So these are the 3 sort of focus areas, and I think for the most part people are trying to integrate technologies right now.
It was Obama initiative. Is Donald Trump likely to kill it?
Oh, we were all very worried about this last year, and I don't know if you saw the news, but Trump had proposed I think it was a 40% cut in the NIH budget. And what they were going to do was they were going to cut university overhead. So right now, Georgia Tech charges a 57% overhead on my grants, and they wanted to cut that to 20%. So that would be devastating, right? So with a concerted effort with Francis Collins, the head of the NIH, and a lot of university presidents, somehow they negotiated a $2 billion increase in the NIH budget instead of a 40% cut. So we're good for this year. So the NIH budget is about $42 billion this year, and it funds the majority of biomedical research in the U.S. And so far, so good. The NIH does have broad support across the political spectrum, and I mean, everybody wants to cure Alzheimer's. It doesn't matter if you're a Republican or a Democrat. So hopefully we'll be okay, but time will tell there. So far, he's kept Francis Collins as the director of the NIH, and that's at the president's request. And then the budget is so far so good.
One more question?
Hi, very impressive talk. It's regarding your tracking algorithm. So you have to use some sort of image tracking, I suppose?
Yes, right.
But then when you do it on the real tissue itself, then how do you track? Do you track a macroscopic soft tissue?
Right. So in the video I showed, and I'll just jump back to a picture here so we're all on the same page here. Let's go with like this one here. Okay. So in this case, what this thing is doing, I'll just pause it here, is there's a microscope overhead. This is just a fluorescence microscope. It's not even confocal or anything special. And it's taking pictures of the cells continuously so that we know where they are. Okay, now those cells are on the very surface of a brain slice. If you want to go below the surface, the only technologies that are available to you are 2-photon microscopy. Okay, confocal can go a little bit lower, like 100 microns, but it's really not that much better. You need 2-photon to really go down 500-600 microns, which is like layer 3, 4 of the cortex. So this is not showing that technology. This is just using fluorescence microscopy. Yes?
This is a question about, so it recognizes or it's targeting soma, but what happens if you've got processes in the way?
Yeah.
You just kind of blow them out of the way? Or?
Yeah, so a lot of people want to record from dendrites out on the distal ends, and that's very difficult to do with patch clamping. There was recently a paper published on what they call a dendrode. It's like an electrode for dendrites, a dendrode. I don't really believe it myself. It was published in Science, but I didn't believe it really worked personally. They only had like one recording. So it's still a challenge. It's still a challenge. Optical tools are pretty good on that. There are tools that optically that can see the propagation of the action potential through the dendrites. Very cool stuff.
Did you say that these recordings were simultaneous?
Yes, absolutely.
So you're able to get 2 patched neurons?
Absolutely. Yeah.
Is there kind of a limit to how close those neurons are?
Okay, so if you're in vitro like this, in a slice, then you can get the tips very, very close together, within 100 microns.
And you don't disrupt the other patch when you're coming down?
Oh, no, you absolutely do. It's very, very important. We have a paper under review in eLife about that right now. What we discovered was that the pipettes have to have certain coordination. So what we do is we find multiple cells, and then we seal onto independently, and then we seal onto them simultaneously. Yeah. So when the delicate business of sealing is going on, you don't want anything else moving, as you can imagine, right? It's kind of obvious. So we let them each find their cells, and then we get everybody parked nearby, and then we go in all at once. Yes, that coordination is very important.
Okay, all right, David, last question.
You sort of mentioned that things got out of the way as you moved.
Yeah.
So you tend to think of tissues being reasonably sort of solid. How solid do you think it is? Is it more fluid?
So the viscosity of brain is it's like toothpaste. And I like to think of it as like a bowl of spaghetti and meatballs. So you're trying to patch onto the meatballs, and the spaghetti strands are kind of slippery and just kind of slide out of the way. We've experimented with the positive pressure, so we crank it up pretty high, more than a bar of pressure, and we can push. Now you don't want to turn it up too high, because what happens is you're spraying the whole brain with intracellular solution. This has got ATP and GDP. It's not good for the brain. But basically, the somas are giant, and they don't move much, and that's how you push everything else out of the way except for those, basically. But in vivo patching is still a challenge because of all this stuff in the way.
Okay, 5 o'clock. Are you going to be around for?
Sure, I'll hang out for a bit. Yeah, nibbles, absolutely.
So let's thank Craig very much for a fascinating lecture.
Thank you.